Hello, and thank you all for attending today's webinar. My name is Dr. Connor Gomes. I am a development scientist with Pion, and I will be presenting to you the exciting new development we have for our subcutaneous injection site simulator, the ECM XR, an artificial extracellular matrix for extended release studies inside the SCISSOR platform. Below you'll see the names of the others here at Pion that were involved in the development process regarding the ECM XR, along with our collaborators at Bath University, Randy Merstein's lab, and our colleague Kate Gridley there headed up a lot of this work, and we want to thank them for their continued role in our development. To start this webinar, we will be reviewing two instruments that Pion is responsible for developing that are used to collect the sets of data that we present within this presentation. The first of which is the subcutaneous injection site simulator, the SCISSOR N3 system. The SCISSOR N3 was a further developed version of the SCISSOR N1 that was released this year. The N3, as expected and will be reviewed, increases the flexibility of the instrument to do 3 independent experiments at the same time instead of just one, hence the name. The second instrument that we will review is Pion's Rainbow R6. It is a fiber optic spectrophotometric device that we will use for in situ concentration monitoring that will be responsible for giving us the pharmacokinetic release profiles and dynamics. After we review the tools of the trade that will be used, we'll take a deep dive into what it takes to actually develop these artificial ECMs, both the ECM and ECM XR. That will involve going over the material characterization of each prototype before and after being used in a SCISSOR assay. We will then go into injections of positive and negative green fluorescent protein, or GFP. The superpositive and supernegative molecules will help us probe the ends of the electromagnetic spectrum and how those molecules act within each ECM. There will be some caffeine injections that we'll talk about. This will probe how polar small molecules act throughout each ECM prototype. Commercially available rapid and basal insulin formulations will be used to try and validate the in vivo, in vitro correlation that SCISSOR is supposed to give. And then we'll last look at a monoclonal antibody formulation with the monoclonal antibody of interest being denosumab. And we'll see how this long-acting biologic looks for a release profile within the ECM and ECM XR. As mentioned, the first instrument we'll be looking at is the SCISSOR N3. We'll be going over the components of the instrument as well as how it was used within these ECM studies. The SCISSOR N3 instrument, which can be seen on screen, again is an expanded version of the SCISSOR N1, where the SCISSOR N1 was essentially just a single one of these chambers with slightly less functionality as compared to the SCISSOR N3. The SCISSOR N3 took that chamber and expanded to give 2 more, and each of these reaction sites will act independently from one another, giving you complete control of each reaction center. In order to understand how the SCISSOR N1 and N3 both work and how we simulate the subcutaneous environment and track pharmacokinetic behavior of in vitro injections to match them to in vivo assay results, we're going to take a look at all of the functionalities involved in one chamber. So to zoom in on the first chamber here, we can see that there's various functionalities and sensors located in the SCISSOR chamber assembly. What we're going to focus on first is this large chamber, the receiving chamber, which normally holds 60 or 300 milliliters of carbonate buffer. Carbonate buffer is the buffering system that the blood uses within our own human bodies. This buffer is pH controlled and monitored via a receiving chamber pH electrode that comes and sits down right in here. While the instrument is monitoring the pH throughout a SCISSOR assay, when the pH gets above a certain set point, it injects a tiny bit of carbon dioxide right behind this ECM cartridge, which can't be seen in this image. Excuse me. That carbon dioxide is blanketed on top, which will then bring the pH of the carbonate buffer back down to the set point. We also want to control the temperature to match the in vivo conditions in the subcutaneous space. So a temperature probe or a thermocouple is placed on the other side of the ECM cartridge. This thermocouple will typically be set to 34 degrees Celsius, and when the temperature gets below that set point, this heating stir plate is then activated to bring the temperature back up to the set point and then turns off again. It will rinse and repeat that for the duration of a SCISSOR assay. The ECM cartridge, as I just mentioned, is the cartridge that will hold the artificial extracellular matrices. In this case, it would be the original ECM and the ECM XR in the following experiments. The ECM cartridge has a porous membrane on these windows that allows for diffusion of buffer and injectate in and out of the ECM space, while hopefully containing the artificial extracellular matrices that we have placed within there. Injection into this cartridge is achieved through this formulation injection port. At the same time, there is an ECM cartridge pH electrode that is located at that injection site that will monitor the pH change from injection back to the pH of typically 7.4 due to the fact that this carbonate buffer will diffuse through the porous membranes as previously mentioned. Lastly, if we are going to actually monitor in situ how fast an injectate is leaving from the injection site through these windows, we need the rainbow fiber optic probe in situ concentration monitor, and that probe will sit right outside the ECM cartridge through this and will be placed through this port right here. That fiber optic probe will then translate absorbance data per time point to the computer to then match to the SCISSOR assay. If this fiber optic probe is not used, manual sampling or auto sampling via auto sampler will take place from this chamber to then be transported for offline analysis in something like a plate reader or a HPLC. So this is what that would look like, the situation where we have a rainbow R6 monitoring in situ concentration via absorbance profiling with these fiber optic probes that are placed within each SCISSOR chamber. So 3 fiber optic probes means 3 separate absorbance profiles for 3 separate experiments. Once again, if these fiber optic probes are not present, there is an auto sampler or sampling port that will take the place of these fiber optic probes for each independent SCISSOR experiment. Each rainbow fiber optic probe has a tip that will be situated within that receiving chamber where the solution will be able to passively move through without any sort of interference. The light from the fiber optic probe will pass into that solution, be absorbed or interact with any analytes there, any scattering particles, any aggregates. It will reflect off of a mirror at the bottom of the probe and back up through the same fiber optic tube or fiber optic line back to each spectrophotometer that is then analyzed with a typical UV-Vis, methods. The gap length of each of the probes can be controlled by exchanging the tip length. The tip length that is used on all of the studies that we used UV-Vis for concentration monitoring, we use the 10 millimeter tip, which has a 5 millimeter gap because the light will then pass to the bottom, be reflected back up to the tip. Through the explanation of the SCISSOR and the rainbow, we understand the use cases that our artificial ECMs will run into, whether it be, interaction with the camera located in SCISSOR, being optically clear for the turbidity sensors, having viscosities and mechanical properties that allow it to be situated within the ECM cartridge without leaking. All these come into play when we think about developing these types of materials. But over all of that, we have to make sure that the biomimicry and the biomimetic properties of our gels match what we're looking for in the subcutaneous space, because if they can't simulate the subcutaneous space, then there really is no point of SCISSOR now, is there? So we look at the human subcutaneous space, which is a collection of proteoglycans, glycosaminoglycans, structural proteins like collagen fibers, as well as a whole plethora of soluble proteins and other structural proteins around. And all of these can act as separate independent reaction sites at the site of injection. And so first off, that biomimicry has to be ensured within the artificial extracellular matrices. So we use like compounds and like molecules to the ones found here, like the proteins and GAGs that I just mentioned. In addition to the biomimetic nature, we have to remind ourselves that these are used as a consumable product and that the ECM cartridges have to have a certain shelf life. The ECMs have to have a certain shelf life, and that allows for formulation scientists to keep them in storage conditions for an extended period of time, which we'll talk about in a second. And then how reproducible each ECM is obviously important, because the whole objective of SCISSOR is to understand the rank order performance of the pharmacokinetic properties in separate formulations. So if we can't have ECMs that act reproducibly over that entire shelf life, then our ECMs won't be too valid over a long time span. So we look to maximize these two things. Lastly, and like I just mentioned, the release and the correct rank order of release is nested within this reproducibility that we look for. If our ECMs can't perform like the subcutaneous space, then there really is no point. The in vivo in vitro correlation is important because the release kinetics and the release profiles that formulation chemists track after an API is released out of the ECM cartridge windows, they have to correlate to what is seen in the clinic and what's actually happened inside a human being for these data to be important for these formulation developers. Lastly, we want our ECMs to be optically clear, like I previously mentioned. So iteration by iteration, as we approach a perfect biomimetic platform, we are attempting to keep the optical properties beneficial for these in vitro assays. For us, that is clear and easily controlled so that we can get a beautiful image of what's going on inside the cartridge, as well as qualitative data with the LEDs. Now, before we get into what we actually were able to achieve here during the development pathway through the ECM XR, which involved revalidating the ECM, we want to look at what work has been previously done by Bath University and Randy Mirsky's lab. Randy was the initial inventor of the SCISSOR platform and this original artificial extracellular matrix. And the way they validated their recipe for this artificial ECM was comparing Genentech supplied monoclonal antibody formulations that are used in the subcutaneous space. Genentech supplied these nondescript formulations to Randy. His lab was able to run them on the SCISSOR in their own artificial extracellular matrix, which is analogous to the model that Pion has available now. And what we're able to see, that the human data and the SCISSOR data, which are the yellow and the green bars respectively, had a much better correlation than the animal preclinical models had to the actual human clinical data. So going forward, we look to match the validation that the ECM was able to go through, as well as compare our results on the ECM XR to these initial ECM results, because such good work had been done previously to validate the performance of this ECM. When initial development started of the ECM XR, we looked to elongate the experimental window that the ECM had previously been validated within, as we just talked about on the previous slide. So initially, we're looking to increase that assay time from now about 1 day sweet spot that most SCISSOR assays are run within, and elongate that for extended release formulations, and have the ECM XR be stable hopefully up to 2 weeks, most likely 1, but hopefully 2. This is most likely going to be achieved through increasing the mechanical stiffness of the ECM, therefore allowing less diffusion of the bulk material throughout any duration. And although we're changing these mechanical properties and changing the formulation of the ECM, we still would like to conserve this 6-month shelf life. We understand that these ECMs are consumables, and the formulation chemists will need a little time before using. In the ECM, this 6-month shelf life is achieved through the addition of a reactive preservative, hopefully due to the underlying formulation or underlying recipe that makes the ECM XR, we have some innate preservation built in that allows for or delays bacteria growth or inhibits bacteria growth. These will both be kept within a fridge or 2 to 8 degrees Celsius in cool storage conditions. The only difference between the chemical reactivity, at least initially at the very start of a scissor experiment, will be during the stability period. Due to the manufacturing practices that make the ECM XR what it is, there's an initial pH range from about 7.2 to 7.7. We had talked about how the ECM cartridges have these porous membranes that allow diffusion in and out of the cartridge for everything except the artificial extracellular matrix. And during the stability period, this pH will equilibrate pretty close to the pH 7.4 because it will be in flux with the surrounding carbonate buffer set at that point. We employed an Anton Par MCR102E rheometer to understand the mechanical properties of the ECM and ECM XR. We use this instrument not only to understand how each manufacturing steps affects the mechanical properties of each material, but we were able to understand the complex viscosity and storage and loss modulus before and after an extended scissor assay to really show the differences between the two models. So a rheometer, for those of us that don't know, is basically a metallic sandwich, and in between that sandwich is your typically aqueous or gel-like sample. The upper plate will either rotate or oscillate, and in order to overcome the force that the sample puts on the upper plate, a torque has to be put on the upper geometry, and the instrument knows how much force it takes to move the upper plate, and therefore it can tell you things about how much your sample is resistant to flow, aka viscosity. With the ECM, before and after assays lasting more than 100 hours, we saw almost complete loss of any sort of mechanical properties or mechanical characteristics within the ECM cartridge. It just further demonstrates that the ECM really isn't tuned for these extended release profiles or injections that are supposed to come out over an extended period amount of time in the subcutaneous space. A closer look at the ECM XR, we can see that we were able to conserve about 20% of the overall complex viscosity. And I know it doesn't sound like a lot, but it's magnitudes over what we were seeing with the ECM while still conserving the chemical reactivity that's found in the ECM theoretically. The storage and loss modulus just sort of paint a deeper picture where we can see that the ECM XR is actually conserving its viscosity by holding on to the sort of solid aspects of its architecture while also still trapping some of the aqueous parts that are more labile. So with the rheology, we were able to confirm the material behavior of the ECM XR elongating the experimental window while also creating a basis set for the ECM. We wanted to look at and ensure that the ECM XR's chemical reactivity was at least analogous to that of the ECM, especially because the ECM, the original ECM, had been previously validated by the Mersenne group for biologics. Caffeine, not a typically subcutaneous injected molecule, but it is a small polar molecule that diffuses in a range of tissues in the human body, specifically passive diffuses. So when we inject this molecule into both the ECM and ECM XR, we don't want to see it be really impeded at all. We want to see ready release and similar release between the ECM and ECM XR. So from this graph, you can see in a SCISSOR N3, we were able to run 3 chambers of ECM and then 3 chambers of ECM XR with that 50 microliter caffeine injection. Barring this one trial of early release, we see similar behavior, pharmacokinetic release, with all 6 of the injections, all of which released 100% release within about 1 day. So just good news for Pion, where the ECM XR, where we look to, make a more a stiffer gel over time, still demonstrates the correct passive diffusion nature, even with the different material characteristics. Once the passive diffusion of a polar small molecule is validated in the ECM and ECM XR, we looked to a slightly larger protein, supercharged protein, green fluorescent protein, GFP, where the superpositive and supernegative analogs synthesized by the Merstein lab at Bath University, and thank you again, injected these GFP molecules into both the ECM and ECM XR. This was a useful way to not only see how these smaller proteins act when released from the ECMs, but it also gave us a chance to probe the ends of the electromagnetic spectrum and how each of the charges would act within an artificial ECM environment. We expect tight complexation of any positive molecules due to the negative environment found within the ECM cartridge. This is a biomimetic in nature, where the large GAGs found, glycosaminoglycans, found within the human subcutaneous space are what are dictating a lot of the charges found there. With the negative molecules, we expect a molecule that is ready released and readily picked up in the receiving chamber, because we expect the negative GFP molecule won't want to stick around and sit inside that negative environment. If we look at the top graph, which is the positive GFP in both the ECM and ECM XR in orange and green respectively, we see that the ECM allows for slight release from the ECM cartridge, where the ECM XR trends lower, almost not allowing any release of the positive GFP molecule from the ECM cartridge. The behavior within the ECM XR is expected because the ECM XR is a very stiff gel that is negatively charged, so we don't expect a lot of passive diffusion out of the cartridge by a positive molecule. The ECM allows for slight release due to the changing environment within the ECM, as it is tuned for more this 24-hour assay regime. So over these longer periods, we get more and more deviation from the norm. If we look at the negative GFP molecules, which are both expected to readily release from both ECMs, the original ECM shows almost immediate and complete release from the injection site, as to be expected due to the electrostatic reasons previously mentioned. The ECM XR looks to actually tightly complex the negative GFP, and this may be due to that the increased stiffness that we found when we were characterizing the material properties of the ECM XR. This smaller, what would be a theoretical pore size, doesn't allow for full diffusion of the very charged negative molecule outside of the pores that are leading to that porous membrane. So overall, we were able to show that the ECM XR does elongate the experimental window, especially when it comes to the positive complexation of molecules, but more probing is still needed to move up in the scale of biologics. We were not able to only probe the electrostatic nature of the GFP injections, but because the ECM and ECM XR are both optically transparent, and the GFP molecule is so fluorescently green, we can actually see how each injection is forming a bolus and diffusing through each prototype. So with the ECM images in orange and the ECM XR images in dark green, the before injection images of both look like analogous materials, which was the objective of the ECM XR development, was to match the same optical properties as the ECM as to not affect any of the scissor related functionalities. With each injection labeled positive and negative, in both the ECM and ECM XR, we have a nice bolus formation with those 100 microliter injections, which is good and was a developmental checkpoint for the ECM XR, where the ECM XR boluses look very, very similar to the ECMs, and we have previously validated the ECM's behavior inside the scissor system. When we look at the 24 hours post-injection, we start to see the real differences between the positive and negative injections, where in both the ECM XR and ECM, the positive injections seem to complex within each artificial matrix more heavily than the negative analogs. This can be seen by the brighter green color in both the images as compared to the negative. Where we can see the largest difference between the two prototypes was that the ECM XR did not allow or did not need full diffusion of the entire ECM cartridge, where the only, I would say, top half of the cartridge is probably utilized for those diffusion of pharmacokinetic behavior. Where in the ECM, the entire cartridge, we can see diffusion throughout the entire cartridge of this positive GFP molecule. This is sort of in support of the mechanical characteristics that we saw before, where the ECM XR has a stiffer architecture as compared to the ECM, which would therefore allow more diffusion through the cartridge. Up until now, the caffeine injection and the GFP injection were great ways to probe the passive diffusion, chemical reactivity, and optical properties of the ECM and ECM XR. And although it's great that we got a bigger understanding of our prototypes, we wanted to probe how commercially available formulations acted within each of the materials. And so to the ECM and ECM XR, we injected both rapid and basal insulins that can be found on the market today. The rapid insulin formulation that we chose is set to release within humans within 2 hours, according to the clinic, and the basal formulation has about 3 to 4 times slower than that at about 8 hours. After injection, you see complete release. So with the light green and the light peach found in the graph, I apologize for the behavior of the standard deviation lines, but the rapid release profiles can be seen in the early parts of the graph with the ECM and ECM XR in the light peach and light green respectively. Both of the overlapping release profiles sort of show the similar behavior as we saw in the caffeine, where the ECM and ECM XR both allow release of molecules that are ready to passively diffuse out of the injection site. When we look towards the ECM XR injections with the when we look towards the basal injections with the ECM and ECM XR, again in orange and green respectively, the ECM shows a drop in the pharmacokinetic release from the injection site in the ECM to a reasonable degree, while the ECM XR shows a very significant decrease in the pharmacokinetic behavior. What this demonstrates is that although they both show and both sort of validate SCISSOR as a rank order formulation pharmacokinetic analysis tool, the ECM seems to be a better model to use in these injections like insulin because of the reasonable decrease in the pharmacokinetic behavior compared to the almost too much of an impact can be seen when using the ECM XR. Lastly, we looked at a commercially available formulation of a monoclonal antibody. That monoclonal antibody is denosumab. The denosumab formulation that we chose to explore within our ECM and ECM XR prototypes releases over 10 days with a total range of 3 to 21. So although the clinical data has that large deviation, we were mainly probing for appropriateness or fitness of each ECM material for this type of injection, a long-term injection and a biologic. So as you can see on the graph with the ECM in green and the ECM XR in orange, we can see that the ECM shows a reasonable release with 100% or complete release within about 100 hours, where the ECM XR exhibited incomplete yet full release after about 160 hours of assay. Now, although both ECM prototypes showed reasonable behavior, the ECM XR was the only one to model that appropriate behavior where we have incomplete release over a large day range. So, in conclusion, Pion and Bath University were able to demonstrate material characterization of the original ECM that was developed and validated at Bath University and Pion's ECM XR before and after SCISSOR assays to show an increased stability period of the ECM XR that elongates that experimental window. The injection of the polar small molecule caffeine demonstrated that both the ECM and ECM XR prototypes show appropriate diffusion characteristics for polar small molecules that would exhibit that passive diffusion and no active reaction sites along the large and multiple reaction sites like the larger molecules would. The injections of both the insulin and positive negative GFP showed how predictable and analogous the chemical reactivity and complexations were within both prototypes, where the GFP was able to further demonstrate the differences between the ECM and ECM XR when it came to the differences in the stiffness of the architecture. Lastly, the monoclonal antibody injection of denosumab revalidated the ECM while further validating the ECM XR for use in these larger biologics that are tuned for extended release in the subcutaneous space. Thank you all for listening to Pion's webinar on its ECM XR development within the SCISSOR platform. And if you have any questions, we will now take them. Thank you.